So we're going from the oceans, not to the mountains, but to space. He's pretty cute, but it's really hard to convince him to go in that suit. Thanks to the organizers, space is truly the final frontier. If I was really cool, I'd have the music playing and James Kirk talking. But as you can see, like any frontier, space is a pretty dangerous place. And when you come down, even after only a 6-month trip, astronauts can't even walk because their muscle atrophy is so severe. It's a concern after 6 months, and a mission to Mars, which would take upwards of several years, is a big concern for NASA and has been for quite a long time. But NASA's also concerned about other tissues, as are we, and we became interested in looking at what happens to liver after spaceflight. It's a key metabolic organ, and it's important for maintaining glucose production as well as overall lipid homeostasis. Our lab is interested in response of liver lipids to oxidative stress. We look at obesity, and we were interested in looking at the spaceflight environment as another model for oxidative stress, and to see what we could learn from spaceflight that we might be able to apply to obesity models as well. So we think that, like in obesity, liver health might be altered in spaceflight, and it might manifest in differences in storage of, for instance, glucose and triglycerides and vitamins, in metabolism of various molecules, and in regulation of liver processes. So to test out our hypothesis, we were able to acquire liver samples from female mice that were flown aboard the very last space shuttle flight, Atlantis, which was flown in July of 2011. It went up for 13 days, and our samples were matched by ground controls. They were age-matched and housing environment and food matched. They're housed in these animal enclosure modules shown here, and so both on the ground and in flight. The mice just floated around in them, and apparently they treated them just like a little rodent wheel, and they just ran around and were very happy up there in space. They were both fed the same type of rodent food bar, which has a composition that's very similar to normal chow, except it's got some different oils in there that prevent crumbling, because they didn't want to have food crumbling up in space. And by telemetry, they had light-dark cycle, humidity, oxygen content, and everything like that all matched. So the first thing we wanted to look at was storage, and we looked at glycogen stores and changes there. And so here's liver sections that were stained with periodic acid shift staining to measure glycogen. The ground controls show, the purple staining here shows that there's quite a bit of glycogen in the ground mice, but it's all disappeared in the flight mice, so they've had a profound loss of their glycogen stores. So that made us wonder if fuel utilization was changing, and if the mice might be actually storing lipids in their livers to use for fuel instead of glycogen. So we wanted to see if more lipid droplets were present in the flight mice livers, and also if that lipid composition might be changing as well. So to look at that, we used Coherent anti-Stokes Raman Spectroscopy, or CARS analysis, of the lipid band, which is a resonant Raman shift at a wavelength, a wave number of 2845 inverse centimeters. And hopefully you can see it. It's a little bit difficult to see, but in images from 3 ground control mice, you might be able to see little bitty red dots in those black images there. And those are lipid droplet signals. And they're very, very small, but I think you can see that in the flight mice, there's a lot more. There's significantly more, and when we do densitometry analysis, indeed, we can see that there's significantly more lipid droplets that we can detect in the livers of these flight mice. So when we did a composition analysis, we first did point Raman analysis, and so this is done on a single lipid droplet. And we did this on multiple lipid droplets from multiple sections and multiple mice, and then averaged them together. And so each of these spectra from ground mice and from flight mice, here in black, represent averages from multiple different experiments. And so here is the lipid band region that I showed you before at near 2850 inverse centimeters. And you can see there's not very much of a difference here. But in this region, there is a big difference in this peak represented by A in the ground mice and C in the flight mice. And comparing with various standards, we were able to identify this band as retinol, or vitamin A, which was present in the diet that was fed to both of the mice. We then used stimulated Raman scattering, which is basically like Raman, but on a very limited range of laser frequencies. And again, here is the lipid band region on a little bit bigger section of tissue. And so you can see that there's not too much change between the ground mice and the flight mice here. But at the region near 1600 centimeters, the retinol region, the ground mice, you can see clearly the lipid droplets, but they've all gone away in the flight mice. So in these two different ways of looking at it, we really do believe we see a significant and profound loss of retinol in flight mice livers. So we wonder what could have happened to the retinol. So I had to learn about retinol and what it does and where it is. And what I learned was that about 80 to 90% of the retinol in your body is actually stored in the liver within a certain type of cell called a hepatic stellate cell. And things can activate the stellate cell. Liver injury activates it. Oxidative stress, which is known to occur in spaceflight, activates stellate cells. And we've been wondering if maybe microgravity itself might activate stellate cells. That's never been looked at before. But what happens when stellate cells are activated is that they actually export all their retinol. So it makes sense that our stellate cells may be activated. So then we were wondering, well, what might happen to the retinol after it's exported? And so we used transcriptomics and pathway analysis to start looking at retinol metabolism. And so all-trans retinol, which is the form that that vitamin A is found in, can be metabolized to visual cycle metabolites, but we found those genes were all downregulated. It can be metabolized to all-trans retinoic acid, but those genes were downregulated. So the genes involved in isomerizing all-trans retinol and oxidizing it towards 9-cis retinoic acid were all upregulated. So we think that the pathway is sort of directed towards 9-cis retinoic acid. That's really interesting because it turns out 9-cis retinoic acid is actually a very potent agonist of a nuclear factor called RXR alpha. So why RXR alpha is interesting is it's very much involved in lipid metabolism by virtue of heterodimerizing with PPAR alpha. So PPAR alpha and RXR alpha together are involved in fatty acid uptake and transport, fatty acid oxidation in mitochondria, peroxisomes, and microsomes, in triglyceride storage, in cholesterol metabolism, particularly biosynthesis and secretion, and in glucose homeostasis. So to see if all of this was happening in our mouse livers, we used metabolomics and transcriptomics and put all the data together and did some pathway analysis. So our metabolomics experiments were performed by Metabolon. We used 6 samples per group with positive and negative electrospray mass spectrometry and GCMS. 299 biochemicals were identified in total. And we found 39 metabolites were identified as significantly changing. 27 were significantly upregulated, 12 were downregulated. There was another 18 that were trending towards significance. And so this was the group that we used primarily for investigation. So when we look at uptake molecules and dietary fats, when you have food coming in, the bile acids in the liver go to the intestines, they emulsify the fatty acids, which then are transported back to the liver and then utilized. So we saw a significant increase in bile acids, cholate and taurodeoxycholate, suggesting that the dietary fats are being more effectively emulsified. We saw a trend towards increase in the dietary fats, although only N3-DPA and linoleate were significantly increased. But there was a trend in the other ones, suggesting these were being taken up. And also our transcriptomics data showed a significant increase in CD36 and FABP2, and so these are genes that are involved in taking fatty acids up into the cell. So these were all increased, suggesting uptake was increased. Triglyceride storage was also increased. We saw a significant increase in glycerol and a trend towards increase in monoacyl glycerol from our metabolomics data. The transcriptomics data showed increases in all these genes involved in triglyceride synthesis. So these data sort of match what we saw in our CARS data with the increase in the lipid droplets. Interestingly, although saturated omega-6 and omega-3 unsaturated fats were all sort of equally distributed in the ground mice, we saw a significant increase in all these fats in the flight mice, but a very significant increase over all of them of the omega-3 fats in the flight mice. And I kind of was wondering why that might be. You know, they're antioxidants, so it could be response to oxidative stress, it could be compensatory. We wondered if they were maybe anti-inflammatory. It could be maybe that they were somehow involved in the PPAR alpha pathway, and they may be regulating or regulated by PPAR alpha. So that was something we wanted to look at as well. We also saw a very significant increase in beta-hydrobutyric acid. So when when fatty acids are undergoing beta oxidation and they generate acetyl-CoA, it generally goes into the TCA cycle for further oxidation. But under fatty acid overload, that's kind of put on hold, and the acetyl-CoA then goes into generation of ketone bodies. And so we do see an increase in in the abundance of mRNAs that go into ketogenesis and this elevation in ketone bodies. We also see an increase in the products of omega oxidation. So it seems like these alternative lipid handling pathways are being upregulated in spaceflight. So we see upregulation of the dicarboxylic acids, tetradecane dioate and hexadecane dioate, as well as glutarate, which is a branched chain amino acid product. So in trying to kind of figure out what this all meant, we sort of went back to our PPAR alpha pathway analysis and Ingenuity Pathway Analysis, and we looked at the targets of PPAR alpha, and I came upon acyl-CoA thioesterases, because we noticed that we had all these increased in fatty acids, and the idea that we potentially had fatty acid overload. And so the ACOTs have been shown to be important under conditions of fatty acid overload, and they offer a fetal cycling in mitochondria and peroxisomes. So they basically will let fatty acids that are in excess exit the mitochondria and peroxisome and go back out into the cytosol, so they can come back in and undergo beta oxidation when the pathway kind of clears out a little bit. So what we think might be happening is that our 9-cis retinoic acid from the activated stellate cells is activating RXR alpha, which is upregulated. That's heterodimerizing with the nuclear factor FXR, which is upregulated, and with PPAR alpha as well. This pair is activating pathways involved in LDL and HDL metabolism, and you can see the genes here that are being upregulated. The mRNA expression is increased. Cholesterol secretion is increased. These pathways mediated by PPAR alpha, such as fatty acid uptake, is increased, and we see, of course, the triglyceride storage is increased, as well as the genes. We see glycerol metabolism is increased, and thioesterase activity may be increased. So ACOT5 operates in the peroxisomes. It is capable of mediating cholesterol biosynthesis, cholesterol metabolism, which leads to bile acid synthesis. The bile acids then go back and feedback, and they mediate FXR activity. In the mitochondria, ACOT2 can directly participate in ketogenesis. In the cytoplasm, ACOT1 is known to directly mediate omega oxidation. And then omega-3 fatty acids have been shown to regulate both PPAR alpha and ACOT1 activity. So it seems like we have this story that's occurring here where we have activation of PPAR alpha and FXR mediated pathways and accumulation of hepatic lipids that are a consequence of stellate cell activation from oxidative stress and potentially microgravity. So another thing that happens with activated stellate cells is that they transform into myofibroblasts and deposit collagen. So we see an increase in TGF-beta receptor genes, as well as a decrease in MMP2. And so we wanted to know if there was also a concomitant increase in collagen fibrils. And so we used Picrosirius red with cross-polarized light, which is shown here in the upper panels, for ground controls and flight mice. And this is a little hard to see, and I'm sorry. We also used 2-photon excitation second harmonic generation imaging, which is shown here in the lower panels, which hopefully is easier to see, and that's the red signal. And so the collagen is shown in the red signal here, and it's the lighter colored signal here. But they both show that there's an increase in collagen fibril deposition in the flight mice. And this was done by rastering across the the liver samples and looking only in the interstices and not in areas around the blood vessels where you would expect to have much more collagen. So by doing that and taking about 30 images per sample, we were able to see a significant increase in collagen per sample in the flight samples. And so we think that this might be a precursor signaling the development of fibrosis. So we think, I've hopefully at least suggested here, that spaceflight alters liver metabolic function, and we think it's via stellate cell activation. We think that the retinol might play a role in activating some of these lipogenic pathways. One thing I didn't talk about here was the potential contribution of stress, because these animals were actually sacked on the ground after reentry. And if you've ever seen an astronaut go through reentry, and the shaking, you can imagine what it does to a mouse. So we're actually submitting a proposal on Friday to try to get tissues from the first mice that are going to be sacked up in space. So hopefully we will be able to get those tissues and then figure out if reentry stress does play a role in these results, or if it is indeed stellate cell activation, and if that's what we're seeing. So as far as consequences to astronaut health and whether a mission to Mars is going to be a big problem or not, it's an open question. When you have lipids in your liver, it's not a good thing, but it can be reversible if you're at the stage of non-alcoholic fatty liver disease. Once you start getting collagen deposition, liver disease can become irreversible, and we're hoping to be able to determine whether a mission to Mars is going to be in this part of the curve or this part of the curve, and whether we can figure out interventions to preserve astronaut health in that case. So thank you.